Good morning, good afternoon, good evening, everyone. You're welcome to today's lecture. In today's video, we'll be discussing about the structure of DNA and RNA. You can recall in the previous class, we discussed about the introductory portion of or introductory section of DNA and RNA, and we have seen the various components of DNA and RNA. So today we are going to be looking at the structure in more details and what are the processes that this nucleic acids undergoes. So we have DNA, which is an example of a polynucleotide, and it is one of the two types of nucleic acids that make up the genetic materials. DNA exists as a double-stranded molecule, which we shall see the figure below, in eukaryotes, but it can be a single-stranded molecule among some prokaryotic organisms. The two helical polynucleotide chains usually coil around a common axis. DNA must undergo a series of folding and superfolding to enable it to fit into the tiny little space that is available inside the nucleus. You know how cell is so small, and you have the component of the cell, you have the cytoplasm and various organelles that are situated inside, and you have the nucleus, which houses the genetic material. So DNA that is found in the nucleus has to undergo a series of folding and superfolding so that the small space that is available for it, it can fit inside that space. So this continuous folding, it gives rise to what we call the genetic material that is found in the nucleus, which is called the chromosomes. So there is what we call a gene, which is a segment of DNA or part of the DNA that contains nucleotide sequence that are capable of producing a protein. And this functional protein, we can say it is been having the information from the gene, which is part of the DNA. So we have four nucleotide units that are found in DNA, which are the oxyadenylate, the oxythymidylate, the oxyguanylate, and the oxycytidylate. So we have a unique characteristic of naturally occurring DNA molecules. They are very long chain of nucleotides, and a DNA of DNA molecule must comprise many nucleotides. This enables it to store sufficient genetic information that is necessary for even the simplest organism. So it enables this long chain of nucleotides enable DNA molecules to store ample amounts of genetic information that will be enough for the organism to carry out its life processes. For example, if you look at Escherichia coli, E. coli, the genome of Escherichia coli is a single DNA molecule that is consisting of two chains of 4.6 million nucleotides. But if we look at the human genome, when we say genome is the complete genetic information of the complete genetic makeup of an organism is what we refer to as the genome. So for the human genome, it comprises about 3 billion nucleotides. Okay. So now if we look at these three structures, A, B, and C, we can see that in the A is the stacking of the two chains. You have the stacking of the two chains of DNA, and you have B, which is the double-stranded representation, and you have C, which is the bit representation of the double-stranded DNA. This is a bit representation of the double-stranded DNA. So for the RNA, which is a single-stranded molecule, look at it. These are the bases. You can see these are the sugar phosphate backbone, and you have the hydrogen bonds, right? Adenine pairs with what? The thymine, right? And but since it's RNA, you have what? Uracil instead of what? Thymine, and cytosine pairs with what? Guanine, right? So also here you can see A and T, C and G, right? but GC or CG bonds, hydrogen bonds are 3, while A and C hydrogen bonds are what? 2. So for the RNA is a polynucleotide just like the DNA, but it exists as a single-stranded molecule, and it is usually produced from the DNA in eukaryotes, but it serves as the main genetic material in some viruses, especially the retroviruses such as the HIV virus. So for humans, the DNA is the what? Is the uh, is the genetic material that give rise to the what? To the what? The RNA. It means it's from the DNA that we can drive the RNA. We can see in subsequent processes when we come to explain the process of transcription, translation, and so on. So, but in some viruses, for example, the retroviruses such as HIV, okay, human immunodeficiency virus, we can say that the genetic material there is specifically what RNA. So the sequence of nucleotide in RNA is determined by the sequence of DNA that is present in the cell, because the RNA is getting from the what? From the DNA. So this will be made clearer when we come to discuss the subsequent processes. So there are some differences between DNA and RNA. We have just discussed one of them, which is what DNA is single-stranded, while RNA is what DNA is double-stranded, while RNA is single-stranded. There are other differences, and we discussed other differences in the previous lecture. So we say that the sugar component of DNA is deoxyribose, but in RNA we have what? Ribose sugar, and the nucleotides are also different by just one in place of thymine monophosphate. DNA is having what? Uridine monophosphate, which is present as explained. So we have these processes of replication, transcription, translation, and DNA naturation, and DNA denaturation, and also renaturation. So when we say replication, it means it's also called duplication. It is a process by which a replica or identical copy of DNA is made from the parent copy. So you have DNA that is producing or making a photocopy of itself to have another DNA. So from DNA to DNA is what we call replication or duplication. And this process occurs whenever a cell is preparing to divide, cell division. All the information, all the component of the cell, all the materials that is present in the cell when there is going to be cell division, it's going to be divided equally, right? You have the parent molecule, parent cell, and daughter cell. So they have identical copies of information and components. So also the genetic material has to be duplicated. So DNA has to be replicated or duplicated in preparation for what we call cell division. So this process must take place so that the new cell will also contain the same genetic information that is present in the old cell. This is how genetic information is preserved and passed from one generation to the next. So there is what we call transcription, which is the process by which the genetic messages contained in the DNA molecules are read and copied correctly to produce ribonucleic acid. So transcription is from DNA conversion or copying information from DNA to produce what we call RNA. So this process occurs whenever the cell or tissue requires a particular protein to carry out a biological or biochemical process. The protein required can be either enzyme, receptor, or other forms. You remember when we said the genetic information in DNA and RNA is being derived from DNA, right? So when we have transcription, it means DNA read its and copy its information to form what we call the RNA. And that occurs when the cell or the tissue is requiring a particular protein or enzyme. So it means the information in the RNA is going to be what translated to protein, which is the functional product that the cell or tissue requires. So we have now to call the translation, which is the process by which the genetic messages that are carried out in the DNA. You know how we are flowing from DNA to DNA, DNA to RNA, now RNA to protein, right? This is what we call central dogma of what? Molecular biology. So we have translation, which is the process by which genetic messages are carried out by RNA and are decoded and used to build proteins, which can be enzymes, hormones, receptors, etc. So we have DNA denaturation and renaturation. The process of separating the complementary strand of duplex DNA is called denaturation. The double-stranded structure of DNA can be separated into two component strands in solution by increasing the temperature. So when you increase the temperature, decreasing the salt concentration by extreme pH, strongly acidic or strongly basic solution, we can have what we call denaturation. So because of the base stacking and the hydrogen bonding between the bases, the double-stranded DNA molecule exhibit properties of a rigid rod and in solution. It is a viscous material that loses its viscosity upon denaturation as well as its biological properties. So when you have denaturation, DNA loses its properties. So the complete separation of DNA strand molecule occurs over a temperature range 5 degrees range. Okay, the heating disrupts the hydrogen bonds in between base pairs, thereby causing the strand to separate. Adenine pairs with thymine, guanine pairs with what cytosine. So when you heat, those hydrogen bonds will be what broken down, and you have these two-stranded molecules now disintegrating, okay, becoming separated. So this is called melting of DNA, and the melting temperature, which is Tm of DNA molecule, depends on the pH and the ionic strength of the solvent. So purine and pyrimidine based composition also affect the melting temperature of the DNA. So the greater the GC content, the higher the temperature and vice versa. So the higher the melting temperature you require. So you remember G and C forms 3 bonds and A and T forms 2 hydrogen bonds. So if you have a DNA molecule that is having G and C components higher, it means you require more temperature in order to bring it to melting temperature. So there are three bonds between G and C, while there are 2 bonds between A and T. We have explained that. And the melting temperature is defined as the temperature at which half of the helical structure is lost. DNA solution absorbs maximally at a wavelength of 260 nanometer, and the absorbance increases as the double helix is separated into single-stranded. You have the denaturation. So this principle is known as what? Hypochronism. Renaturation is the opposite of denaturation. It is the process of reassociation of the separated DNA strands or any 2 strands of DNA that are complementary in base pairing. This process takes place only when the appropriate physiological temperature and salt concentration are achieved. Renaturation is also called hybridization. So let's look at some importance of some synthetic nucleotides. Synthetic nucleotides are used in the chemotherapy of cancer and viral infections. Modified synthetic analogs of purines and pyrimidines and nucleotides have useful application in clinical medicine. These components are used to treat cancer because they can prevent DNA synthesis due to their toxicity to the enzymes involved in DNA synthesis. Some of these synthetic nucleotides are also used to manage AIDS patients because they are toxic to an enzyme required for DNA synthesis in the virus. For example, azithromycin (AZT) is a potent inhibitor of reverse transcriptase in HIV. Zidovudine is another drug for HIV control. Its active component is 2'-azido-3'-deoxythymidine, which is AZT inhibitor, and AZT inhibits HIV replication. So AZT is not used alone, but in combination with other drugs with different mechanism of activity. The combined therapy is called highly active antiretroviral therapy, HArt. So, and this is some simple questions that you can easily attempt, and also this brings us to the end of these lectures on structure of DNA and RNA. Feel free to refer back to the course material, watch the video, ask questions in the forum, and we shall be able to discuss. Okay, thank you very much. See you in the next video. Thank you.